Bla 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 bla. Bla 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 bla. Funkar det? Jag pratar med producenten. Ska jag stänga? 2. Och sen är du färdigpratad. Nu är det. Okej. Yes. Okay. Yeah, so you're all welcome to this seminar by Dr. Herwig Schüler, who is Associate Professor at the Karolinska Institute and a group leader there. Herwig, he started with his undergraduate studies in biology, biophysics, and chemistry. And in different places, Frankfurt, Uppsala, Stockholm, and then he did his PhD at Stockholm University, studying actin and actin-binding proteins. Very interesting subject for us. Anyway, then Herwig continued to work abroad. He came then back to Sweden and the Karolinska Institute 2008, and is since 2011 a group leader there, using a combination of different biochemical, biophysical techniques and X-ray crystallography to study enzymes ADP, ribosyl transferases, and also developing selective inhibitor acting on these enzymes. So I'm sure you will tell us many exciting things about that. Thank you for this. And please. Okay.
Thank you very much, Al, for the invitation to speak and also for the introduction. So this talk will be about ADP ribosylation, and a key compound in this process is a molecule that most students in the life sciences know under the name of NAD, or nicotinamide adenine dinucleotide. And most people probably know that the reduced form of NAD, or NAD+, takes electrons from the food sources that we burn in our bodies, fat and sugars and proteins, and then transfer these electrons to the electron transport chain, which then drives ATP generation in the mitochondrial membrane. In ADP ribosylation, NAD has a completely different role. NAD is being cleaved into nicotinamide and ADP ribose. And this ADP ribose is then transferred to proteins to elicit all sorts of effects. So it's a post-translational modification, very much like phosphorylation, methylation, and acetylation, which are much more well studied. So what are these enzymes that catalyze this reaction? The most famous ones and most well studied are certainly these bacterial toxins. We have Clostridium, diphtheria, pertussis, and also Pseudomonas toxins that ADP ribosylates proteins in our cells to get access to a replication competent niche. So in simple terms, they use this to turn their host cells into a bag of food. ADP ribosylation is also catalyzed by sirtuins, these histone deacetylases. So what they do is they first ADP ribosylate acetylysins, and in the second step, they eliminate the acetyl group together with the ADP ribosyl group. So these are actually also ADP ribosyl transferases. And finally, we have, in human, we have 5 exoenzymes that act on the cell surface or outside the cell, and we have a total of 18 intracellular enzymes that all characterize by ADP ribosylation. So now in the first part of my talk, I will tell you a little story that we just finalized in my group about the structure and the catalytic mechanism of an ADP ribosylating toxin of Pseudomonas. And I hope I will have time to go a little bit into the other work we do with the human PARP enzymes, the human proteins or enzymes that catalyze ADP ribosylation. So about the toxins, this shows you the evolutionary relationship of all ADP ribosylating toxins. And you can see here that we know quite a lot about the structure and the functions of these cholera and pertussis toxin-like enzymes. And we also know a lot about the clade down here. These are the Clostridium toxins and related toxins. But this clade up here, we knows definitely nothing about the structures, and we know very little about them. So we started out to study them, and it turns out that this clade here has one thing in common. They are all activated once they reach the host cell. They are activated by 1433 proteins. So the 1433 dependent ADP ribosylating toxins. Pseudomonas is certainly the most medically interesting pathogen because it is the reason for hospital-acquired infections. And this is associated to multi-drug resistance and biofilm formation, which makes it really hard to target these bacteria with drugs. And they are very problematic in burn wound patients, in organ transplant patients, and in cystic fibrosis. So many people with cystic fibrosis, or the majority, eventually gets infections by Pseudomonas, and it is often lethal. And then we have several other related species or families of bacteria which also have these 1433 dependent ADP ribosylating toxins, and which are interesting from medical viewpoint. So what also characterizes all these bacteria is that they very early in the infection process make contact with their host cell. So here we have the bacterium on top, and this is the host cell. And they establish this contact with something called the type 3 secretion system, which is a protein complex forming a needle to inject toxins into the host cell cytosol. And depending on the strain of Pseudomonas here, these are 1 out of 4 toxins, and they vary a little bit in content depending on strain. So we have mostly XOS, which then forms a complex with 14C3. Essentially all strain secrete also XOT, and then we have 2 other toxins that occur. And this is, in some strains, this is essential for infection. In other strains, it's not essential for infection, but the activity of these toxins is essential to disseminate later on and invade tissues. So it's certainly an important medical problem. So to study these toxins, we needed to establish a method to measure their activity. And what we did is we use a version of NAD that the only difference really to NAD is this little acino group here that makes this entire group fluorescent. And the fluorescence is sensitive to the local environment. So when we cleave off nicotinamide, then the fluorescence goes up. So what we do is we add the toxin in complex with 14C3 and this acino NAD. And when we then add the substrate protein, in this case RAS for XOS or CRK for XOT, then the substrates get modified, ADP ribosylated, and we get a fluorescent signal. So if we just increase the concentration of NAD and measure this reaction, then they get faster and faster until they reach a plateau. So it follows Michaelis-Menten kinetics here. And this experiment shows you that if we vary the content of epsilon NAD with regular NAD, then the Km value, so this is a midpoint in this reaction, does not change very much. So in other words, the toxins don't use our marker molecule quite as well as regular NAD, but almost as well. So now we screened a small library of compounds. These were known inhibitors of ADP ribosyl transferases, and checked whether one of them would inhibit XOS. And it turns out we found one molecule here that inhibited XOS quite well, and this just shows you the work underlying the determination of the Ki value, or the IC50 value was about 19 micromolar. So this is not a very potent inhibitor, but it's a start, and it's a molecule that we now could use for further studies. Now, XOS is extremely difficult to produce. We do this in bacteria, and this shows you a regular purification gel where we have passed a lysate of bacteria over an affinity column, and we get out this little peak here. And this is the purity. It's not very impressive. And most of it actually ends up in so-called inclusion bodies, which means the bacteria inside the bacteria, they don't fold into a functional protein, and the bacteria take care of them by putting them into inclusion bodies. And you can see even here, it's very little protein compared to all the background proteins in there. So we tried to refold this unfolded material into native proteins, and it doesn't work. So we were thinking maybe we can make use of this natural activator 14C3 and co-express the 2 proteins together and get better yields. So for that I will have to tell you a little bit more about 14C3 proteins. So these form dimers that are bowl-shaped like this, and inside this bowl you have 2 binding sites, one on each monomer, for phosphorylated proteins, for phosphate groups. So what they do is they bind to over 2,000 different phosphorylated protein and regulate their activities. So this can be simply by sequestering the protein. It could be by sequestering it so it cannot bind to different binding partners, or it could actually bring two proteins together so they form a complex. And in human we have seven different isoforms of this 1433. What we also knew already about the interaction of XS and 1433 is that the C terminus of the toxin binds in this phosphate binding site without itself being phosphorylated. So several groups have studied this, and this is a crystal structure of just a peptide that corresponds to the C terminus of the toxin. And we see how it's bound in this phosphate binding site, and the interactions made are hydrophobic in nature. So the green area here is a site on 1433 that's hydrophobic, and where we have the interactions with the toxin tail. So this shows you also that we have these hydrophobic residues that interact at that site completely conserved in all 1433 dependent toxins. And for comparison, I'm showing you here a fifth sequence of a toxin that is not dependent on 1433 but otherwise has the same activity. So it's not conserved in that one. So from on the onset of our studies, the model people had looked something like this. We have an inactive conformation of XS that is then captured by 1433 by binding at this site, and then somehow the conformation of the active site changes, so you get an active enzyme. And we wanted to simply be able to understand this in detail so we can possibly make a drug against this. And now we want to co-express the toxin with 1433 to get higher yields, to be able to do crystallography. So first we need to find out which of these 7 isoforms is the best one to use. And it turns out 6 of them bind approximately the same to the toxin. And the 7th one is the sigma isoform, binds half as well. And the explanation is that sigma is the only one that doesn't efficiently dimerize, so you only have the monomer. So that's probably why we see this effect. But the 2 isoforms that bound best to the toxins were the beta and the zeta isoform, and the beta was absolutely the best, so we used that one to co-express the 2 proteins in E. coli. So the first problem we encountered is that when we used this vector to transform bacteria, we never got any colonies. And well, essentially we got some colonies that always contained mutant proteins. Either the toxin or 1433 was mutated. And we did that also in the presence of our inhibitor that we found in the agar, and never got any wild-type colonies. So we could say that the toxin, when co-expressed with 1433, gets toxic to bacteria. And our solution to that problem was that we introduced 2 mutations in the active site of XOS, and these we knew by sequence comparison. And then we could make this protein. And in the case of XOT, we could make the wild-type protein by co-expression, presumably because it doesn't modify the same proteins in E. coli. So now I've shown you these gels before. Now we get this amount of yield. It's a totally different story. And we get, now it's very overloaded here. We still get some impurities with it, but we have a lot of soluble protein now. Now we crystallize this with robotics, and this shows you a 96-well crystallization plate where we dispense the protein, and we have 96 different buffers for crystallization. And these 3 wells on top here, they are on a little shelf where we can put 3 different concentrations of the same protein. And then we put this into an automated imager, and we just wait for what happens. And we get crystals immediately. This shows you these needle-like crystals here, and they don't diffract. So that was a disappointment, of course. So we had to take a step back and focus on the purity of these proteins. So we introduced a second step, or a third step actually, an ion exchange step. And you can't really see it too well here, but we get the protein in different complexes from this ion exchanger. So here's one, and then it goes down a bit and up again. Here's the second peak. And when we use this protein, then we get better crystals. Now we can get some diffractions of the crystals in the X-ray beam, and finally we could solve the structure of this. Now what we also did is we analyzed these fractions from the ion exchanger by a technique called SEC-MALS. So size exclusion chromatography, and then multiple angle light scattering, which can tell us the size, the exact size of these proteins, or the protein complexes that we get from the ion exchanger. And it turns out we get two different peaks here with a small difference in molecular size. And one of them is a heterotrimer. So it's 1433 with one toxin molecule bound. And the second one is a heterotetramer, 1433 dimer with two toxins bound. And when we looked at the active site, it turned out that the C-terminus of 1433 was lying right across the active site, blocking the active site. So in other words, this was useless for us. We wanted to understand how does the active site work, right? So we had to essentially start over from beginning and construct vectors that didn't express the C-terminus of 1433. And when we did that, we finally got our structures, and now we know exactly how NAD is bound. So this is actually done with a variant of NAD that's non-hydrolyzable, because it has a carbon atom in this site where you'd find oxygen in NAD. And so we understand all the side chains reactions and exactly how NAD is bound. And this is the structure with our little compound that we found in this compound screen, and that inhibits the toxins. And what we also saw is that other peaks from this ion exchanger that indicated larger molecular weight complexes were also present in the crystal packing of our protein crystal. So this shows you this complex here that we can find again. And what we think is happening is that it's present both in the crystals and in solution. So what we think is that there might be some autoregulatory mechanism. So we get larger complex formation that somehow regulates the activity. So what about the C-termini of XOS and XOT that I've been talking about initially? The sequence that was already known to bind to 1433. Well, it turns out the first one, this is the same as the peptide structure I showed you before. And it looks exactly the same in our structure. There's almost no difference in how it binds to this hydrophobic site in the phosphor peptide binding groove of 1433. And to our surprise, there was a second peptide here, and that was the extreme C-terminus. So the second peptide bound to the other monomer in the 1433 dimer in pretty much the same way. So this is the first and well-known LDLA motif, and in the very C-terminus, all these toxins, or most of them at least, have a second LDLA motif that nobody had paid any attention to, or at least it's not mentioned in the literature, and that one bound to the other site. So now, of course, the question was, what's the meaning of having these two different binding sites, and can we find out what their specific functions are? To do that, we made constructs of the toxin with a GFP on its N-terminus. And then we recorded fluorescence anisotropy curves, so fluorescent polarization, where we make use of the speed of rotation of larger versus smaller molecules. And then when we titrate the binding partner, 14C3, then we can get these bindings curves. And we can see that if we use the entire toxin with both of these LDLA sequences, then we get an affinity of roughly 40 nanomolar, which is quite a tight protein-protein interaction. If we cut off the last one, so we're left with only this one, then we still get 70 nanomolar binding. If we use none, we still have 200 nanomolar binding. So cutting off both of these sequences, we still have sub-micromolar binding of these two proteins. And that was a huge surprise. And this is just to show you that the sequences by themselves just linked to GFP can also bind with sub-micromolar affinity. So without showing you any more details, we weren't able to find out what these two separate sequences do and what their functions are. But we think it may also have to do with regulation of the toxin once they are inside the host cell and bound to 14C3. So this result here indicated, of course, that there must be another binding site between the toxin and 14C3 that could give us this very high affinity. And that's indeed what we found in the crystal. So this is the regular LDLA sequence bound to the phosphopeptide binding site that has been known before. And now we have a large interface here. And if you take these two proteins and open them up like this, then you can see what this interface consists of. It's the white region here is hydrophobic residues that stick to each other. And on each side, we have positively charged against negatively charged residues that form salt bridges, and the opposite on the back here. And zooming in on this hydrophobic binding site, I've just sketched out some of the interactions in more detail. So these are really strong interaction in a hydrophilic environment. And then we got lucky because around the same time, another group published a crystal structure of 14C3 with a small compound bound to its surface. And that's the one here in blue, called NV1. And it turns out this compound is just on the edge of this binding site that we had just discovered in our crystals. So we ordered this compound, and we ordered a few others that look a little bit like it and are even larger and even more hydrophobic. And then we tested what these compounds do to the activity of the toxin. And the best one was this one that we call 1704. And it interacts with 14C3 to break this interaction with the toxin, and it inhibits the toxin. So again, the potency is very low. It's not a great inhibitor, but this may be the beginning of a whole new story about antibiotics, namely that we can now inhibit the toxin by blocking the interaction with its activator. So that's a very attractive way to go forward here. But of course, we still didn't know how this toxin gets activated. So we were thinking, what in this toxin can link this binding event to 14C3 to the active site up here? Is there any amino acid that can relay this information to the active site and activate it? Elicit some sort of conformational change. So without going into the details, we mutated a whole bunch of residues here, and none of the mutants had any less activity than the wild-type toxin. And then we thought, well, if we can bind the toxin with no tail to 1433, maybe we're thinking the wrong way. Maybe we don't really need 1433 binding for the activity. So what we did is we looked at similar toxins that were not 1433 dependent and where the structures were known. And we took the one that had the best match with our structure, it's called V's toxin, and then we took its C-terminus and replaced it for the natural C-terminus of XOS. And we get activity. So we get activity. Here is just glycohydrolase activity where the NAD is turned over with no substrate, and here is the activity when we add the natural target protein RAS. And in either case, we get activity that is not further stimulated by 1433. And again, this follows nice Michaelis-Menten kinetics. So what we see is really true. So this means that the ADP ribosyl transferase domain does not need 1433 binding to get activated by a conformational change. It already has what's needed for activity. So at this stage, my postdoc, Peter Hornjak, he got a brilliant idea. He said, let's just forget about the literature from the last 20 years and do this ourselves. Do we really need the C-terminus of the toxins to bind 1433? We already showed that we had interaction with a construct that didn't have the C-terminus, but would it be active? That was the big question. So I showed you initially how the wild-type toxin co-expressed with 1433 was toxic to bacteria, and now we have the same here. If we use the mutants, we can make bacteria. Here we induce expression, and we can still form bacterial colonies. But if we use this small segment in the co-expression vector, then we get toxicity. So this indicated the toxin was indeed active, even though it didn't have this C-terminal sequence. And this is just this fluorescent assay showing you the increase in fluorescence when we add the natural substrate again. And this is using a different fluorescent nucleotide or NAD analog that we can observe in an SDS page gel. And here is the modified RAS protein, and this is some auto-modification of the toxin. And we don't have that if we add the mutant protein. So again, nice Michaelis-Menten kinetics. So and then we were pretty close to the answer, because now it seemed that we have activity in this part that doesn't need the tail of the C-terminal tail of the toxin to bind to 1433. Sorry. So 1433 binding was needed for something unrelated to activity. So what we did is we looked again at dynamic light scattering, which can measure the aggregation of protein. So the black dots here show you what happens if you add the complex of 1433 and the toxin. It has a very high melting temperature, so it's stable. And the white dots show you how unstable the toxin is if you don't add 1433 to them. So in other words, binding to 1433 prevents aggregation of XOS. And this actually shows you how this little compound that blocked the interaction can be used to show the same thing, that if we add the complex, this is how it melts, so it's the same curve as up here, the stable protein. If we add DMSO, which is the solvent that our inhibitor is in, it destabilizes a little bit because we think that this hydrophobic interaction is already broken a little bit by the solvent. But if we add the compound that blocks this interaction, then we get a very unstable protein, just as if we hadn't added any 1433. So this leads us to this model. In conclusion, we think that if XOS is expressed, in our case in bacterial cells, or if it is squirted into the cytosol of our host cells from bacteria, then it can aggregate. And of course it does that no matter whether you have the C-terminus present or not. Now if the C-terminus is present, then it can be bound to 1433, and 1433 can pull active toxin out of these aggregates. So we believe the answer to the question is 1433 is simply a chaperone for ADP ribosylating toxins that are 1433 dependent. So that is the end of this part. And now I'd like to show you just a little brief summary about our work on human ADP ribosyl transferases. So what I showed you before is the transfer of this one ADP ribosyl group to acceptor proteins. In the human enzymes, this can get much more complicated. We can get polymerization of this ADP ribose into long and branched polymers. And these are used in different ways. So this is just symbolizing the enzyme, the writer function, where we get either mono ADP ribosylation or poly ADP ribosylation. And for each class of ADP ribosylation, we have specific proteins that bind, recognize these, and elicit downstream functions. And then we have erasers, glycohydrolases that can then specifically recognize either mono ADP ribosylation and cut it away, or it can recognize these chains and shorten them or remove them. And that in turn regulates lots of different processes, including transcription and DNA repair, and maybe most importantly is the ADP ribosylation of histone proteins. And we have one project where we try to study this process by just using these histone tails that you probably know are sites of multiple modification by all kinds of enzymes. And we fuse these histone tails to GFP. Oops, the P is in the wrong place. And then we can study how these proteins interact, and we hope in the long term to be able to solve a structure of this complex between the enzyme and these modified histone tails, or the recognition domains and the modified histone tails. But what's sorry, this slide is definitely wrong. See if I can show it like this. What's interesting about these PARPs also is that they are important drug targets for cancer drug development. So this enzyme PARP1 recognizes single strand breaks in our DNA. And that leads to recruitment of the repair machinery, and this break is repaired. If we now inhibit PARP1, then the single strand break turns into a double strand break. And now we need a whole new machinery of DNA repair enzymes to take care of this defect. And that is usually done by homologous recombination. And one pathway that is key here is the BRCA pathway. And if we have defects in the BRCA pathway, then these defects never get repaired and the cells die. So it turns out that many cancers, especially breast and ovarian cancers, have defects in these BRCA genes. And these two studies in 2005 showed that we can selectively kill cancer cells with BRCA defects by inhibiting PARP enzymes. So now we have a whole bunch of clinical trials going on with PARP inhibitors, and 3 compounds have already been approved by the FDA for treatment of breast and ovarian cancer. And we had solved all these different crystal structures of PARP enzymes, in total 10 different PARP enzymes. So we had all these proteins, and we knew how the structures looked and how the active sites looked and how these inhibitors would bind to them. And we said that we wanted to analyze how does inhibition of PARP1, the clinical target, affect all these other enzymes. Doesn't that lead to effects that we don't want in a drug? That's what you would expect, right? And it turns out, now here we screened a whole library of 200 compounds that looks very similar to PARP inhibitors, and we find that most of them inhibit more than PARP1, that inhibit at least the subclass of PARP enzymes that are most homologous to PARP1 or even other enzymes. This was just done with a biophysical assay, and then we established inhibition assays, enzymatic assays, and looked at least the 7 clinical compounds, the ones that were in clinical trials, and made the entire profile over this entire PARP family and checked how the IC50 values looked for these different enzymes. And this is just one example here showing you that PARP1 is the enzyme that's inhibited best by this particular drug, olaparib. But all of them are, essentially all of the PARP enzymes are in some way affected. So that led us into trying to make our own selective inhibitors for PARP enzymes. And that is basically important so we can study their functions. So we made inhibitors that we know work in cells. They have the expected cellular outcomes, and we have crystal structures that show how they bind to the enzymes. And we distribute those to researchers interested in digging deeper into the functions of these particular PARP enzymes, answering the questions, what happens when we inhibit them specifically and not in context with the whole family? And of course, can we use those as drugs? Do these more selective inhibitors make better drugs? And a second, sorry, I should have switched back to the full screen, of course. This is just a second example dealing with another PARP enzyme called PARP14, where we have 2 different types of inhibitors here. One that blocks the entire binding site, and another one that uncovered a whole new binding pocket for inhibitors, which we think can be very useful to make selective and potent drugs. So this is my last slides. I'd like to acknowledge my team here. This is the people in my team at present. Tobias Karlberg is crystallographer who solved essentially all the structures I've shown you today. And Anja Tuchel is a technician who is extremely skilled in crystallization. So she can look at these protein drops that I showed you and know after the first experiment what she has to do to get crystals in the next experiment. And these are my 2 postdocs. So Katja works on the histone tail binding project, and Peter worked on the toxin project. And we have funding from Cancerfonden, the Inga-Britt und Arne Lundbergs Forskningsstiftelse, Stiftelse för strategisk forskning och Vetenskapsrådet. So with that, I'd be happy to take some questions.
Thank you, Harry, for this very interesting talk. So, comments or questions? Alf?
Yeah, really very nice and interesting. as I didn't know about these enzymes before, and that was very, very interesting indeed. So you were doing the screening of the PARP inhibitors. What specific technique were you using for the screening? You mentioned you'd use some biophysical technique.
Right, so no, I think it's this one now. So we can just quickly go back to this heat map. So the question was, what technique did we use to screen all these compounds? So we used a technique called DSF, differential scanning fluorimetry, where we simply look at a protein domain while it unfolds in a temperature gradient. So when the protein unfolds, the hydrophobic core gets exposed, and then we can bind compounds that have fluorescent properties when they bind there. And then we get these transition curves very similar to what I've shown you in the light scattering experiments as well. So this only works with singular domains, and we only used the catalytic domains of these proteins here, and that was important to get away from. We want to use catalytic activities in the full domain context of the enzymes. So this was, of course, an important study because it for the first time showed how many different enzymes these compounds inhibit. But the second paper I showed you here, these transition curves are actually done with enzymatic activity.
More comments or questions?
And how did you measure these enzymatic activities? What technique?
It's essentially the same way we measure the activities for the toxins. We have a variant of NAD, the substrate, with a modification that gives you the signal in the assay.
Protein for that, or?
No.
Well, I mean, this is of course relative, but
yeah.
How much would you need, you think?
Well, for a complete binding curve of an inhibitor done in triplicates, we would need something like 0.1 milligrams of enzyme or something like that.
Sven?
So this NAD molecule, it's very important for the energy metabolism in the cell. And here you put it on proteins, you can put large chains of it on the protein. Isn't that a problem for the cell to get rid of all these important molecules?
Yes, definitely. So that's what you're asking is a whole subfield of our PARP field, and I'm not a specialist in that. But yes, there are definitely interactions between, for example, DNA repair on one hand, where you have lots of NAD being used up by marking the sites of breakage and formation of these chains, and energy metabolism on the other hand. And one use for PARP inhibitors was actually for, is it called ischemia? I don't know how it's pronounced, but where you have essentially so much DNA repair going on during apoptosis of the cells that they get necrotic. They use all energy for making poly-ADP ribose chains, and the tissues get necrotic. And that's actually one use of PARP inhibitors as well.
More questions or comments? So when you look at the structure here, you do X-ray crystallography. Would you, there have been a lot of recent development in cryo-EM and image analysis. Could it be something that you would see with that technology compared to the cryo? Well, compared to the X-ray?
Yes, well, I guess it depends on the question you're asking. We wanted to study the exact interactions of all these compounds with the core of the proteins, which is typically quite small. So for us, it was sufficient to use that single domain. But in some of our cases, we have multi-domain enzymes. And to understand the regulation, we definitely want to look at how do they interact with each other. And cryo-EM is a good method for that. And actually, also in the toxin project, we try to study these larger complexes that I've shown you with cryo-EM.
Okay. Yeah. More comments or questions? If not, Herwig, another warm hand.
Thank you.
And this was the last seminar this semester. Please don't go, because we have to do some additional information here. I think Edin fixed the camera and come down, and Sandra, please come down, and Peter. During the walk towards here, we are going to say a little bit about next semester. We are going to move, as you all know, and that means that we're going to have seminars in September and maybe the start of October, but then nothing more than until the end of February or something like that. So please expect, and maybe Alf is going to give a seminar in September, if possible, and run also. So there will be some seminars in September, but not more, because then we are focused on moving the stuff to another location. Right, so with these words, we conclude this semester, the seminar series, and thank you, Harry, to come and have the final talk. Now, it is, I'm going to switch to Swedish. Is that okay? Yes, Louisa? Okej. För att vi ska kunna klara av att hålla de här seminarierna, så det här är faktiskt ett teamwork. Jag syns ibland, Peter syns, men faktum är att många som gör ett väldigt viktigt och bra jobb här, det är bland annat producenten, den som spelar in, och idag är det Edin, men han och Ingmar delar på det här ansvaret, och det gör de på ett mycket bra sätt. Och sen är det någonting som är oerhört viktigt, och det är just det här med att se till så att seminariehållarna skickar in en titel och lite text, och sen se till så att annonseringen kommer ut på de olika institutionerna, se till att det här registreras i diarie, eller inte diarie, men i, vad heter det, flödet. Ja. Och där har vi turen att ha Sandra Hedberg, och som då hjälps av en annan tjej som heter Lena Finnas. Och Peter och jag skulle vilja då som en liten tack för det här insatsen då ge en liten present. Det här är till dig och Ingmar. Ingmar. Ja, Ingmar. Det här är till dig, Sandra. Och så får Lena också. Så, och så tycker jag vi ger dem en applåd. Och nu så, med det här nu så avslutar jag alltså den här terminens seminarieserie. Och ni är varmt välkomna. You're all very welcome to this short semester series next semester. Have a nice summer.
Thank you.
So, I got a little idea, but let's wait with that.